All right, hello everybody. I hope y'all had a good weekend. For those of you that are fathers, I hope y'all enjoyed your Father's Day. I'm going to go ahead and jump right into this. We're in Lab 6 on enzyme activity. It's related to chapters 8, 9, and 10, especially with an emphasis on 8 and 9 in terms of the fact that we talked about enzymes. Purpose of this lab or the objectives of this lab I have on the next page. It's after completing this exercise, you should be able to describe what an enzyme is and how it works, and describe how the following factors affect enzyme activity: substrate concentration, temperature, and pH. Those are 3 of 6 that we went over in lecture for chapter 8, okay? So if you need some review on that, you can also look at chapter 8 lecture. Now, when we talk about enzymes, you have to know what the purposes of an enzyme or what its function is. The function of an enzyme is to catalyze chemical reactions in living organisms. Specifically, we focus on cells since it's a cell molecular biology class. We talked about how these enzymes often require energy in the form of ATP. Now, ATP is not going to be a factor in today's lab. What we're going to be focusing on is enzyme activity, and you would in person be performing what we call enzyme assays. They're often called enzyme assays, where you measure the rate of enzyme activity, and then you also measure the rate of enzyme activity by varying certain factors. Now it says here, enzymes can produce products such as proteins, or can produce breakdown products such as fatty acids from triglycerides. As we talked about before, okay, in order for a chemical reaction to occur, we need reactants. However, when you're talking about enzymes catalyzing chemical reactions, okay, some of those reactants, or at least one of those reactants, is going to serve as a substrate. Keep in mind, I said this in lecture, okay? Not all reactants are substrates, but all substrates are reactants, and you'll see that again today when I give you the formula. When we talked about how enzymes, okay, they're proteins, so their shape is directly related to their function. For an enzyme, though, we talked about there's specific regions in an enzyme. In this case, they're showing you two. The one I showed you in lecture was one. But there are specific regions within an enzyme, okay, that have a shape that complements the shape of the substrate or substrates. We call these regions of the enzyme the active site. So here they're showing you on this enzyme, we have two active sites. And it's these active sites that are specifically going to bind to the substrate. It's also at this active site where a chemical reaction is going to occur, okay, and it's going to result in the enzyme converting the substrate to a product. The enzyme you work with in today's lab, okay, it actually requires 3 reactants. One of those 3 reactants, glucose, is a substrate. And the enzyme is not going to produce one product, as in this example here. It's going to produce 2 products. Okay, so it varies as far as number of substrate reactants that function as substrates versus number of products that are produced. But this gives you a general idea of what we're talking about. Now, this information on the slide is what I just talked about here, so I'll let you look at that information. I'm going to jump ahead. Also in today's lab, you're going to study 3 of 6 factors that can affect the rate of enzyme activity, now, or altogether inhibit an enzyme from becoming active or staying active. So if you look here, each enzyme has its highest activity, meaning it causes a chemical reaction to occur the fastest under specific environmental conditions. So listen to me carefully. The enzyme you're working with in today's lab is an enzyme that is found in cells of our body, and it helps our cells metabolize glucose. Okay? Therefore, this enzyme prefers a temperature of 37 degrees Celsius. This enzyme prefers a pH of roughly 7.2. Okay? So those are 2 factors you need to keep in mind. Now, what concentration it requires, it wasn't given in the lab, and I didn't look it up. Specifically what concentration it needs. But you can see here the reason why I'm mentioning our body temp and the pH within your cells, as these are 2 of the 3 factors you're going to be studying, okay, that can affect the rate of this enzyme's activity. Now, any factor that affects the interaction between an enzyme and its substrate will affect enzyme activity. And the way you're going to determine if enzyme activity is being affected by any of these 3 factors, okay, is simply by measuring the amount of one of the 2 products that are being produced. So we talked about temperature. Okay, with temperature, the enzyme I said you're working with today is commonly found in our cells. That means it prefers a temperature of 37 degrees Celsius. That is the optimal temp for the enzyme. But keep in mind, I said this in lecture for chapter 8. The optimal temp for an enzyme, okay, doesn't mean that's the greatest amount of product the enzyme can produce. The optimal temp for an enzyme means that the enzyme will produce the optimal amount of product that a cell needs. The enzyme might actually be able to produce more if you change the temperature, but that doesn't benefit the cell. So the enzyme needs to be at its optimal temp so it'll produce a concentration of product that the cell needs. Now what happens though is when you expose enzymes to temperature outside of that optimal temp, say you expose the enzyme to very low temperatures, that slows down the rate of enzyme activity. Okay, so when enzymes are exposed to low temperatures, their rate of activity decreases. However, when enzymes are exposed to very high temperatures, what happens then is the enzymes denature. They unfold. So in the case of today's enzyme, there's going to, one of your three experiments, okay, has to do with temperatures. You will expose this enzyme to 4 different temperatures: 0 degrees Celsius, room temp 22 degrees Celsius, body temp 37 degrees Celsius, and 65 degrees Celsius. Now in table 2 in the lab procedures, I've given you data, okay, associated with the amount of product the enzyme is producing over a duration of 120 seconds or 2 minutes. And how much product the enzyme is producing is measured actually every 15 seconds. So you'll see these absorbance values increasing. And I'll go over more what these absorbance values mean in a bit. Okay? Well, 65 degrees Celsius is a very high temp. And what's been found is that when enzymes are exposed to very high temperatures, okay, they denature. Therefore, they lose their shape. They no longer function. So high temps inhibit enzymes by inactivating the enzymes. Enzymes are no longer active. So when you look at that data, you got to look at the data and you got to first look to see, is the enzyme producing product? And if it is, how much product is it producing? Then you got to compare the different temperatures and determine, okay, did any of these temperatures inactivate the enzyme by denaturing it, and therefore it's not producing any product? It's inactive, it's been inhibited. Do any of these temperatures slow down the rate of enzyme activity? In other words, they're producing product, okay, but they're not producing a lot, at least during the first few intervals that are measured. Okay, and then keep it in mind, okay, if the enzyme is producing more product at a temp that you didn't expect it to produce more product, well, you got to keep in mind the optimal temp for the enzyme has to do with the enzyme producing the correct amount of product that the cell needs, not a maximum amount of product. pH will be another factor you're going to determine how it affects enzyme activity. This will be recorded in table 3. I've given you data in table 3. The enzyme you're working with in this lab has been exposed to 4 different pHs. The lowest is 3, the highest pH is 10. So again, you first want to look at the data I gave you. I give you absorbances that are measured at 15-second intervals for a duration or total of 120 seconds or 2 minutes. You want to look at the absorbances and see if you see an increasing amount of absorbance occurring, and then you want to compare the absorbance values from 15 seconds all the way to 120 to see if there are any differences. Okay, that way, as I said, okay, the greater the absorbance value, the more active the enzyme is because the more product it's producing. So you'll look at pH. Now the way it works for pH is that if enzymes are exposed to pHs that are too high or too low, it could cause the enzyme to denature and therefore not work properly. So you can read this information here. The last factor you'll study, which is actually the first part of the lab procedures, is substrate concentration. Okay, we talked about this. This is one of the 6 factors that affects enzyme activity. Simply put, enzymes can't produce enough product if they don't have enough reactants, especially the substrate. Likewise, if you give an enzyme too much of a substrate or too much of the substrate as well as any additional reactants, what will happen is the enzyme will reach its maximum activity. It'll reach a point of what we call saturation, and it can't produce any more product. Even if you keep feeding it more and more substrate, it reaches a point where it says, we can't work any faster. This is it. This is the most we can produce. You can give us all the more substrate you want. Nothing's going to happen. So what's going to happen is you're going to expose this enzyme, okay, to 6 different concentrations of its substrate. And then there are 2 other reactants that will automatically be present in the solution for the enzyme, okay, to act on that reactant, which is a substrate, and start producing product, okay. And what's going to happen is in the pre-lab, the very last pre-lab question, it's going to actually ask you a question to calculate data that you need to transfer to part 1 of the lab procedures. So the last pre-lab questions actually have to do with the first part of the lab procedure, and it's setting up a serial dilution. So if you look at table 1, going left to right across the table, notice the concentrations of substrate that you're going to be giving to the enzyme, okay, have a difference of 2-fold. And I'll let you figure out what I mean by that from there. So the enzyme you're working with today, it's called glucose oxidase. Okay. If you recall, I said they often name proteins that function as enzymes with the name that ends in these 3 letters, ASC. The first part of its name comes from its substrate, which is down here, glucose. So all 3 of these are reactants. Hopefully I can write a little better today. Okay. All 3 of these are reactants, but only one of them is acted on directly by the enzyme. Only one of them is bound at the enzyme's active site, and that's glucose. So glucose is the only one of the reactants that actually is a substrate. However, in order for this enzyme to catalyze a chemical reaction producing our 2 products, okay, it needs these additional reactants. So there's 3 reactants, but only one serves specifically as a substrate acted on by this enzyme, which is bound at its active site. Glucose oxidase, as it says, it's an enzyme found in human cells. And in vitro, in a test tube, as we say, this enzyme is used to help measure blood glucose levels for those that are diabetics and other disorders. Now, what happens with this enzyme is that this enzyme will take these 3 right here and produce a total of 2 products. Okay. It'll produce 2 molecules of gluconic acid and 2 molecules of hydrogen peroxide. So it produces an equal ratio, 2-to-2, of the 2 products. In this lab, scientists developed an experiment to determine the activity or the rate of activity of the enzyme glucose oxidase by measuring the amount of the product hydrogen peroxide. Since this enzyme produces an equal ratio of both products, if you can just detect one of the 2 products, okay, you get a good idea of not only is the enzyme active, but how active. Okay, so it's not just an issue of enzyme activity, it's also an issue of the rate of enzyme activity. So when you look at the 3 tables that I've given you with data already recorded, data that you normally would have collected yourself, as I said, you first want to see, was the enzyme active? Do I see absorption values, positive absorption values after 15 seconds? And then every 15 seconds after that, up to 120 seconds, do I see the absorbance value increase? Because the absorbance value is being caused, okay, in part, not totally, but in part, the absorbance value is being caused by this one of 2 products, hydrogen peroxide. Now, how is this? Scientists discovered that in solution, if you take these 3 reactants and this substrate, glucose, okay, and you take this enzyme, glucose oxidase, and you combine them together with the addition of 2 chemicals. These 2 chemicals are known as phenol and 4-amino-antipyrine. Okay. If you take those 3 reactants and combine them with the enzyme glucose oxidase along with these 2 chemicals, what will happen is that this one of two products, hydrogen peroxide, when it is produced by this enzyme, glucose oxidase, the hydrogen peroxide will react. The hydrogen peroxide will react with these two chemicals, creating a pink color. And what happened was scientists discovered that this pink color absorbs light best at a wavelength of 510 or A510. So the way it would have worked in lab is you would have been given a flask with just the substrate, or a flask with the substrate glucose. The other reactants, they're already available, the oxygen and the water, in the air and in the solution. Then you would have been given a separate flask with the enzyme glucose oxidase, except the enzyme would have been pre-mixed with these two chemicals. Therefore, whenever you add the substrate glucose to the enzyme solution containing glucose oxidase, and the glucose oxidase hasn't been inhibited, what will happen is the glucose oxidase will produce 2 molecules of gluconic acid and 2 molecules of hydrogen peroxide for every 2 molecules of the substrate glucose. And every time the hydrogen peroxide is produced, it will react with these two chemicals that were pre-mixed with the enzyme, producing a pink color. Therefore, if you look down here, thus the rate of increase in the absorbance of pink dye will be your measurement of glucose oxidase enzyme activity. What happens is, for most of the experiment, when you first add the substrate to the enzyme, you would then place that solution mixture into a spectrophotometer preset at an absorbance of 510 or wavelength of 510. Then you would hit a play button so that the spec would take an absorbance reading at 15-second intervals. The first 15 seconds, it'll take an absorbance reading. Second 15 seconds, it'll take another absorbance reading. And if the enzyme is converting the substrate to product, what will happen is at every 15 seconds, the spec, measures the absorbance, a light absorbance. What will be happening is the enzyme is taking reactant and produce a substrate glucose and producing more and more product. And this more and more product is reacting with more and more of these two chemicals. Therefore, what happens is the intensity of the pink color increases. Therefore, your absorbance increases. So you'll see your data, the numerical values for absorbance slowly increasing, which means the intensity of the pink color is increasing because the enzyme is producing more and more product. The catch with this lab, though, is that once you combine the substrate, with the enzyme's not waiting for you to take your sweet time and put the sample in the spec to start measuring how much product the enzyme's producing. This is a moving reaction or a rolling reaction. Once you put the substrate with the enzyme is taking the reactants or the substrate glucose along with the other reactants and producing product. And it's going to keep producing product as long as there is available substrate and other reactants. So it's a moving reaction. So what you see is for those reactions where enzyme activity is not inhibited, your absorbance values keep increasing. So what we tell students is, if you had done this in person, we would have told you only add the substrate to the enzyme one sample at a time. So you would have taken glucose, the substrate, added it to a cuvette containing your enzyme. You would have inverted it a couple of times to mix it, wipe down the side, put it in the spec as quick as you can, hit play, and the spec would start reading right away. And if the enzyme is active, the amount of pink dye produced, every 15 seconds, the intensity of that pink color would increase. Therefore, your absorbance values would increase. So if you look here, this is just repeating what I said. You can see here what it tells you. You would have measured the absorbance at 510 of the amount of pink dye being produced every 15 seconds for a duration of 120 seconds or 2 minutes. But keep in mind, the pink dye is only produced if one of the two products, glucose oxidase, produces hydrogen peroxide is being produced. So that's how we indirectly determine if it's working. Your lab report's straightforward. Same usual bit with the pre-lab questions and answers. The post-lab is divided up into 3 parts. Listen carefully. Part 1 has to do with taking your data in table 1 of the lab procedures, creating a scatter plot, then asking it to do a linear regression analysis, and then asking it to give you the linear equation, y equals mx plus b. Okay, that's pretty much questions 1 and 2. Under 2, though, it tells you, look at your value for the slope, represented by m. The value of your slope, okay, tells you the rate of enzyme activity. In other words, how active was your enzyme? The higher the slope value, the more active the enzyme was. The lower the slope value, the less active the enzyme was. So when you give me answers about what concentration of substrate the enzyme prefers, what concentration of substrate is the enzyme most active as, make sure you also cite that slope value. It's got a numerical value. It's going to be a similar pattern for part 2. This is where you're going to take the data I gave you in table 2 under lab procedures. You're going to create a graph under part 2 post lab with the 4 sets of data I gave you for temperature, and then it's going to ask you similar questions, and you're going to use the slope again to justify under what temperature is the enzyme most active or least active. And then part 3, you're going to take your data I gave you in table 3 of the lab procedures. You're going to create a graph, just like it tells you in part 1, but you're going to be using the data I gave you for pH. And then what you're going to do is you're going to analyze that data, focusing on the slope to justify at what pH level is the enzyme most active and least active, okay? Now, real quick, what else I want to show you is the lab itself. Go down to the lab procedures. So you can see here under lab procedures, you can see here it's giving you the time intervals every 15 seconds up to 120 seconds or 2 minutes. And then across for table 1, this has to do with substrate concentration, but it says glucose. Well, what is a substrate for the enzyme glucose oxidase? It's glucose. So they give you the name of the substrate. And then notice the concentrations of the substrate glucose increase twofold, left to right. And notice the absorbances I gave you. They start off very low, but they slowly increase, meaning that the enzyme, when given time, will produce more and more product using the substrate glucose and the additional reactants. If you go down a little further, this is table 2. Table 2 has to do with temperatures. Here are the 4 temperatures you would have performed the experiment at, but notice here in the left column, the time frame is still the same. Okay, but you can see here now you have to not only look to see, do you see an absorbance value at 15 seconds and does it increase? Okay, but you also need to compare them between each other, okay, to determine when is the enzyme most active. Well, you're going by that absorbance rate, whichever one has not only the highest absorbance rate at the end, but you're also looking for how quickly did it start producing product. It may not produce much product at the end because it used up all the reactants and the substrate glucose. Okay, so keep that in mind. And then here's table 3 for part 3. Table 3 for part 3, here's the pHs that you expose the enzyme to. They're actually buffers, but each of these buffers maintains the pH given in the table, but notice the time frame is still the same. Okay. So go over the information, make sure you figure out, okay, what data represents, okay, the x-axis, what data represents the y-axis, okay? The reason for creating these graphs is to help you analyze your data so you can extract conclusions, okay, from your data that's given to you in these 3 tables. Okay, and keep in mind, when you give me your answers, you should be able to cite some numerical data, okay, that you're extracting from the graph that you created using this data. So the purpose of these labs, okay, is to teach you how to perform an experiment, to teach you how to collect your data, to teach you how to start analyzing your data, and to teach you how to draw conclusions or extract conclusions from your data. This one's probably one of the simpler, more straightforward ones to teach you how to do this. Okay. As far as due date and time, that's all posted under assignments for Lab 6. All right. Y'all take care. Bye.